Hi everyone, welcome to the Space Science webinar series hosted by the Center for Space Science at NYU Abu Dhabi in collaboration with the UAE Space Agency. Our today's speaker is Dr. Dirk Schulze-Makuch. He's a professor of planetary habitability and astrobiology at the Technical University Berlin in Germany. He's also an adjunct professor at Washington State University and Arizona State University. He got his PhD at the University of Wisconsin in Milwaukee. Then he worked in industry for a while. He was an assistant professor at the University of Texas at El Paso. While he was also a faculty fellow at NASA Goddard Space Flight Center. After that, he moved to Washington State University, where he was a full professor. He got the Frederick Wilhelm Bessel Award from the Humboldt Foundation for extraordinary achievements in theoretical biology in 2011. Since 2013, he's a professor at the Technical University Berlin, where he leads the astrobiology research group. He publishes in astrobiology centered around planetary habitability. He has nearly 200 scientific articles and has written 10 books. Today, in addition to his research, he's also going to talk about his recent book. So Dirk, welcome to our center, and the stage is yours.
Thank you for having me. And thank you for giving me the opportunity to talk a little bit about deserts, Mars, and life in the universe. Let's start when we look at this with Earth. And if you think about Earth, we think about it as habitable for us, Homo sapiens, at a temperature of 0 to 40 degrees C, pressure of 1 bar, on a solid medium we live, and we are heterotrophs, meaning we eat the other guys. But Earth is also habitable for a lot of other organisms, like the great white shark, which is in some way similar, but can take a much higher pressure and lower temperature and lives in salt water. But Earth is also home for extremophiles like Arcidianus infernus, that can that's optimum living temperatures 88 degrees C, and it's a chemolithotroph and lives under a very acidic pH. And there's not only microbial extremophiles, which we're usually referring to, but there's actually multicellular extremophiles like the crucian carp that lives at very low oxygen conditions and can take living in a pond or lake where there's no oxygen at all for about a month or two. The Pompeii worm can live at temperatures up to 105 degrees C, or how I call it, Conan the tardigrade, which is probably the most extremophilic macroscopic organism that can have tolerances in temperatures from absolute 0 to plus 151 degrees C. Can take space conditions as pressure to up to 6,000 bars, up to 5,000 gray radiation tolerance, and has can do cryptobiosis and hydrobiosis. So it's amazing in adaptation. And that we have to keep in mind when we look at the extremophiles on our planet. For example, with the temperature, the upper limit right now is held by Methanopyrus candleri. It can grow at 122 degrees C. That is, of course, at higher pressure when water is still liquid. The survival range is probably even higher, but not much higher, because then the molecules of life will eventually break down. The absolute maximum is probably around 150 degrees C. If you look at the lower limit, that is very difficult to define, because most of the organisms, microorganisms, for example, just become dormant. We definitely know, we work in our lab a lot with Plantococcus halogryophyllus, that it can grow at minus 15 degrees C. We can also go a little bit lower with some other organisms. And there may be still some metabolic activity down to -39 or -40. But this is not completely confirmed. And again, then it's very difficult to define. So metabolic activity in principle only ceases entirely when the aqueous cell plasma freezes. So it's a little bit subjective where you put really the lower limit. The pressure range of life is pretty high as well. Most famous are at the deep hydrothermal vents. There's the pressures up to the 89 megapascal. And the main obstacle for life is not really how high pressure you can get, but the pressure differences, which is a problem. So only sudden changes. Low pressure, Martian pressure seems to be fine, that organisms can still survive. The main problem is then desiccation. So we're coming now more closer to the conditions on Mars, and most important there is water activity when we look at life. So water activity is about, where life is still possible, about 0.6. So 0.605 is for saturated sugar solution for fungus. Some more recent, maybe in 0.585, but it's about there. However, we have to take it with a certain grain of salt, because I worked in a liquid asphalt lake in Trinidad, and we measured a bulk water activity of 0.49, and we still had active microbes. But they lived in micro droplets of water within the hydrocarbons. So you have to really think about at what scale you measure. There's also water activities can fluctuate daily and seasonally, as they do on Mars. And organism may be just active once a certain threshold is overcome, around 0.6 probably then. So otherwise, they may be just dormant. So there's a little bit flexibility certainly there. When we look in the deserts, we have also to look at the salinity's limit of life. We know that some archaea and halophilic bacteria can grow at 35% sodium chloride solution. We have to realize that life at high salt concentrations is energetically expensive for microorganisms. We don't actually know where the limit is. It may be reached like in waters, like in the Don Juan pond, which is has a lot of magnesium, calcium chloride solution there. So we don't actually quite know it, but organisms can take a lot of salt. In order to investigate further, I founded the Atacama Desert Dry Limit of Life project. And you see, we put a lot of investigators together. That was funded by the European Research Council. And we wanted to see how far life can go as an analog to Mars. And in order to do that, we have to go to the driest places on Earth to see how dryness affects habitability and the dry limit of life. And most suitable for that, we found, is Atacama Desert and the dry valleys of Antarctica. Now, there are some kind of differences here. Of course, Antarctica is much colder, but actually there's less relative humidity in Atacama. And the geochemical processes are very similar. And we focus on the Atacama Desert because it's easier to get there too. Antarctica is always a bit of a problem, logistics and otherwise. So we want to see what are the lessons from the driest deserts on Earth, where land dwellers would have to adapt to desiccation, high salinity, and UV irradiation. What would be that limit of that adaptation, and what is that limit on Earth? So we started off with a hypothesis, and that is a cemetery in Yungay. Humans couldn't survive there for very long, and asking us, are microorganisms slowly dying in Atacama's core desert in the hyper-arid part, or is it at least a temporary habitat? So we sampled along a moisture gradient. This is here at the coast that is most moist, and it's getting less moist. And then you get here into the hyper-arid area. And a particular, also famous, is Yungay. It's one of the driest places on Earth. And you see here that it looks very much like on Mars. Actually, if you look from some of the rover pictures from Mars, you see exactly the same. Very much the same. Of course, you don't see people there. Like we looked here at some nodules, and we also dug some holes to further investigate for possible microhabitats. We looked at the salt concentrations because salts are a concern as well. And as you see, we dug some holes, and actually it's getting so dry that we actually have perchlorates here in the hyper-arid region, just a little bit below the surface. And this is increasing distance from the nearest coastline. So that is close to the coast, and here it's getting more and more hyper-arid. We found quite a bit of biological diversity, again, here from the coast to the hyper-arid area. And you see that the diversity is decreasing, but we still have quite a bit of diversity. We look at metabolites to indicate microbial activity. And you see there's much more, again, we go on here, this is close to the coast, as most of the metabolites we find here. But we actually find quite a bit of metabolites still also in the hyper-arid area. Then we looked at internal and external ATP. We separated that. Internal ATP is thought to be still within the cell. External ATP in the really dry desert, it can actually survive quite a bit outside. And looked at both of those values, and you see that as in general, it goes down. Where here at Yungay, 20 to 30 centimeter deep is a little bit of a maximum still. The phospholipid fatty acids go down. The amount that you find, the diversity you find, that makes sense too. As you're getting drier, the diversity drops. As does the biomass altogether. And in the hyper-arid area, then between 1,000 and 10,000 cells per gram sediment. That's the basic lowest you find. We looked also into internal and external DNA. We use the separation methods to do so. And what you see here, again, going from the coast to inland, over here is this two pools of external and internal DNA overlap very much. So there's a lot of turnover. While you have in the hyper-arid areas here, you have quite a bit of separation. You don't have much turnover. There's a specific pool of external DNA, probably microorganisms that were blown in from the ocean and died there under the conditions in the hyper-arid area. And those that actually still make a living and active, but a small part of it only. We put all this information together in a manuscript and wanted to make the case that there's still active microorganisms in the hyper-arid core of the Atacama Desert. But we still didn't make the case. We sent it to the proceedings of the National Academy of Sciences and got in the first attempt rejected and said, well, we have to do better. And then we decided, okay, we include metagenomics. And what you see over here is in our metagenomics, over here, this is the IRAP values that shows at 1, there would be no reproduction. And you see here that organisms actually reproducing, and we caught them in the moment basically of reproduction and could show that. We also found viruses, and there the argument is viruses would not affect dead microbes, only those that actually active life cycle. And we obtained a 3-year record. And with that, we could make the case that there is a transient habitat. And we published then, we got the paper then accepted in PNAS. And that was in 2018, and since that we have more than 100 citations on that paper. So if we kind of look a little bit back and see if you go into a desert, what happens, it is the following. If you're in an arid area with 25 to 200 millimeters rainfall, we found soils and lithic habitats colonized by cyanobacteria, chlorophytes, and heterotrophic bacteria in the top few centimeters. We developed biological soil crusts, and we have still a highly high species richness, really. But as you're going drier and drier, the colonization patterns become patchier and a gradual decline of cell abundance and species diversity. You also don't have the biological soil crusts anymore, or they become discontinuous. And you get these hyperlithic communities like here. This is a quartz rock that I turned over. So this was here the underside. And you see that there are cyanobacteria growing on the underside, which is a great survival mechanism from them because it's more dense there. And they are also protected from the UV radiation, but still get enough light through the translucent rock. And then eventually you get to hyperaridity with a mean annual rainfall of about 1 millimeter or less. So this means it rains maybe once a decade there. Even the hyperlith cannot do it anymore. Microbial respiration is questionable, and we get to the lowest microbial biomass that is really measured on Earth. So then the only organism that still can survive is actually those inside the rocks. And you see here some pictures of those. This is an endolith at Sala Grande. That is an endolith near Yungay that we measured. You see here's the cyanobacteria inside. You see here cytonomine, that is a pigment indicative of cyanobacteria as well, that living in the salt rocks and actually living using deliquescence. So they can take the water directly up from the atmosphere. And that is a real great adaptation mechanism, because if you are in the Atacama desert, in the hyperarid core, where it doesn't rain, you still can get your water directly out of the atmosphere. And the process is really hygroscopicity. It's the same thing when you have some salt. It's still morning at Abu Dhabi, right? So if you had your salt in a salt dispenser, and usually you have some rice grains inside. If you don't have the rice grains inside, the salt will get clumpy because it attracts the water from the atmosphere. And that is the same principle over here. So the microbes using halite and the hygroscopic property of it. And you know, not in the Atacama desert, but in some other deserts, you actually have some lizards that use this kind of same principle and have a certain hygroscopic grooves where they can attract water. So when we look at the picture, then there is a predictable sequence of ecological transitions that occur on land as conditions become increasingly dry. You see over here, we have aquatic ecosystems that corresponds to the Noachian period on Mars. Then we're getting drier and drier. And so we have the arid, then getting into the hyper-arid areas with endoliths and hyperliths. And eventually at the very end, we get into the hygroscopic niche where you have the endoliths that use the hygroscopic properties of salt to make a living. So, so if you look at Earth and Mars and life on it, we see a tale of very different planets, because Earth is an ocean planet. Very early on, Mars was probably an ocean planet too, but now it is a land planet. So, so we have now a well-preserved geological record of land environments on Mars. So we have to look, how do you survive in such an arid land environment? And if you compare it with Mars today and in the past, we see that, well, the Atacama has about 1,200 precipitable microns. Today on Mars, it's still quite a little bit more arid, drier, but there have been times of high obliquity And that comes pretty close what it is now in the Atacama. So, you know, in the Atacama, life can still barely hang in. Whether it can still be on Mars, it's kind of questionable, but highly interesting to find out. But if we find out, we can have certain conclusions from our research. First of all, the habitability rules of land environments are fundamentally different from aquatic environments. Critically, land dwellers rely on atmospheric sources of water. So we do not expect to find running water on Mars. Nowadays, it does not happen. Otherwise, you may have a groundwater eruption or something like this, but you still have amount of water vapor in the atmosphere. Terrestrial deserts are a vivid example of life that life can exist long after the disappearance of aquatic environments. That's highly relevant to Mars. And there's a predictable sequence of ecological transitions in Earth's desert with increasing dryness that also apply to Mars. So life on Mars need not to become extinct with the disappearance of water, and the Martian habitability window might have closed much later than it's currently thought. And maybe it hasn't even have closed yet. We published that, Alfonso Davila and I, in our 2016 paper, The Last Outpost of Life on Mars, the possible last outpost of Mars. So how does this affect any kind of mission? Where should we send the next Mars mission and search for life? So my point of view, what we should do is actually go to the southern highland of Mars, where you have the chloride salt deposits, and look there for life. I would even go so far to use this as an analog, which we have the Yungay salar. You see here the salt nodules, and there in those rocks, there was cytonomy and the microbes that made a living there in the driest area of the Atacama Desert. And something similar we would expect on Mars too. So I would go that far and say, you know, the mantra of NASA, when you search for life, look for liquid water, is in that way not the best way to go. But I would say when you search for life on Mars, look for the salts, for the hygroscopic salts. And halite, of course, is a hygroscopic salt, sodium chloride. So if you have those, then you that's the furthest you can go, at least from the Earth base, to still find life. And there I would look for life on Mars. Okay, let's go just to make a little bit of a step further. And I mean, of course, we are all excited to find life, microbial life, but we would even get more excited about finding complex life. So the questions that we ask ourselves, can we find a second Earth, a second biosphere with plants or animals like in this nice painting here? Well, right now we have more than 4,000 exoplanets and are still counting. And could there be complex, perhaps even intelligent life out there? There was a famous book by Wharton Brownlee in 2000, the rare Earth hypothesis. Most astrobiologists know that. And they put the hypothesis out that the evolution of complex life requires an improbable combination of astrophysical, geological, and biological circumstances. Thus, the evolution of complex extraterrestrial life is very improbably unlikely to be extremely rare. So we actually beg to differ, and William Baines and I put another hypothesis out, which we call the cosmic zoo hypothesis. We say that key innovations during the evolution of life on Earth have been achieved multiple times and using different biochemical pathways on the path toward complexity, yet with the same function. So the function is the important thing. So if stable habitable planets are common, life on those planets will develop eventually complex forms. So we published a book about it, The Cosmic Zoo, Complex Life on Many Worlds. It was published in German as well. And asking ourselves, are humans a galactic oddity or even freaks of nature, or would you expect intelligent life on other planets as well? So what we want to ask is, what is unique about life on Earth? And would life on other worlds evolve the same way if a tape of life would be rewound? Would we get the same results? So how do we test the cosmic zoo hypothesis? Like I said, the important thing is the function. And you have certain functions like imaging vision, where you have a very different kind of organism evolved independently from each other, or powered flight, where you have some raptors, insects, bats, even some fish that can fly, evolved independently in the many paths toward that innovation. So when we look at it, we distinguish between three major paths toward innovation. A critical path model, where the major event or innovation requires preconditions that take time to develop. So it is basically that, like filling a bathtub with water, eventually all the conditions are fulfilled and the bathtub will flow over. Random walk model is that the major event or innovation is highly unlikely to occur in a specific time step. And the likelihood does not change substantially with time. It's like if you roll with a dice 5 6s, you can roll very long. Eventually, you might be lucky, but we cannot really predict that. Another way to innovation is a many path model. So the major event or innovation requires many random events to create a complex new form, but many combinations are possible. It's like in a poker game. 4 aces will win, a royal flush will win too. So there's many different possibilities how you can win a poker game. So there's many biochemical ways to get there. And I want to show you, if we can show that all the major transitions or key innovations are many path events, then we likely live in a cosmic zoo. This does not mean that we end up with animals and plants like on Earth, but organisms that in their functionality will be animal or plant-like. So I have to see for a moment where I am with the time. Okay, so good. I also want to show you a few selected transitions. The first one, origin of life. Well, the evidence points to the origin of life occurring between 4.2 and 3.5 billion years ago. The recent evidence points to the origin of life on hydrothermal events, but that is not proven. The underlying problem is that it's very difficult for the organic macromolecules to come together, and we don't really have a good idea of it. Obviously, that is a little bit of a weakness because we do not understand really the origin of life yet. We don't know whether it's a many path process. For example, in the many path process, it could happen that there's several different origins of life, just the strongest was selected and has survived. So we don't know for the origin of life whether that's many path process, a critical path process, or a random walk event. Let's get to another major transition, the use of light. Photosynthesis evolved from mechanisms that were meant to protect organisms from light, and particularly from harmful UV radiation. And from Earth's biochemistry, now we know that there's very different ways to capture light. Chlorophyll photosynthesis is most common, but light can also be captured by rhodopsins or carotenoids and turned into chemical energy. So if we look at bacterial rhodopsin-based light capture, is chemically completely different from chlorophyll-based captured. So there's many paths to harvest light. So if you think now being on some other planets, you know, there may be different ways how to do it, not with chlorophyll, but with bacterial rhodopsin or with carotenoids. Major transition, endosymbiosis, or the evolution of eukaryotes. Eukaryotes evolved by endosymbiosis, and this is a frequent event, and that's still occurring today. It's actually occurring between all different types of species, even between archaean animals. And endosymbiosis, we know, led to the invention of mitochondria, of chloroplasts, the nucleus as well, and possibly other organelles. So since it's so common and between all different kind of species, it's clearly a many paths event as well. Let's look at multicellularity. We're getting now closer to where we are, or where we are on the eukaryotic tree of life. And if you look at the tree, there's many different multicellular forms, which are probably originated from colonial organisms. And if you look at those trees, so there's sometimes in the same family, there's a multicellular organism, there's a single cellular organism, and there's sometimes that organisms then can switch between those. And one of the most amazing ones are myxobacteria, actually. And because they can switch between single cellular and multicellular lifestyle. So what is for us a major leap, it seems to be quite easily be accomplished by life. So we call it also a many path event. Let's jump a little bit ahead and look at intelligence. Well, we are not the only intelligent species on our planet. There are cephalopods, birds like crows, whales, dolphins, and primates that are also intelligent. And intelligence is really a needed asset for predating species. So a wolf has to be smarter than a deer that it hunts because it anticipates a prey's move and coordinates a hunt with its fellow wolves. The deer just has to be sensible enough to smell the wolf and get away, but the wolf has to be much smarter. So there is basically a reward by natural selection to be smarter. And so we ended up with a lot of different intelligent organisms. And if you see the cephalopods, the octopus is also very smart, but it's not related to us at all. So a lot of intelligent species. So the call comes to be more difficult when we say about technological intelligence, because our species is the only one that it can modify the environment to its needs, colonize nearly any habitable stretch of land of our planet, and domesticate fellow animals. And we invented fire tool, language, and the heat engine. And we are the only example. This has happened only once in the whole history of our planet. So since this only occurred once in one species, we cannot say whether this is a many path, a critical path process, or a random walk event. So if we summing up, then it can be shown, or that we show in more detail in our book, that all major transitions are many path events, except for the original life and the rise of technological intelligence. That means if the origin of life is not extremely unlikely, that would mean that we are living in a cosmic zoo, with a lot of different organisms, also animal-like and plant-like, probably. But if it's extremely unlikely the origin of life, we could be rather lonely. This we cannot decide. And with a cosmic zoo, we call it cosmic zoo because the other problem, of course, is technological intelligence. We don't know how common that is or how often that will happen. So if we're honing on Earth 2.0 with all the exoplanets, we will see what we find out. Right now, how we detect exoplanets is by indirect methods using planetary transits, radial velocity, or astrometric observations. The problem right now is that we cannot observe the planet and the star separately. And that will be soon, or hopefully soon be done, like with a space telescope like this, starshade called, where the space telescope and starshade have to be exactly aligned. And when we can have at least a pixel of the planet by itself, then we can actually get already more information out. We may be able to measure average temperature and pressure. We may identify whether there's ocean, continents, and polar ice caps on that planet. Determine atmospheric compositions. Are there gases consistent with life? We may be even be able to detect massive chlorophyll-based vegetation like on our planet. And this is something that gives us hope. This is, for example, Titan, which has this kind of reflectance showing there are some liquids. It's not water, it's liquid methane, but nevertheless, you see actually the effect with a space telescope. So the next frontier would be from 1 pixel to mapping an exoplanet to see whether we have a green planet or a desert planet. And what we most would need to do, of course, are site visits. I always tell my students that if you do astrobiology, you have to get close and personal. Just to orbit or look from afar, you cannot do much. The closest here would be Proxima b, but it is 4.2 light years away, about 1.3 Earth masses. It's around an M star, but M stars flare a lot, so it's it would be a pretty harsh environment with a lot of X-ray fluxes and solar flares. And of course, in order to get there, even with the most optimistic projection, will take more than 40 years. So what we should do is use as a testing ground our solar system, where there's enough or very intriguing possibilities, microbial cloud life, Venus possibility. There has been a lot of emphasis on this lately. Near surface life on Mars, subsurface ocean life, possibly Europa, Titan versus liquid hydrocarbons on the surface, and maybe very exotic life if it exists. And Jupiter's moon Europa is very especially interesting in this regard, because in our hydrothermal friends we find complex life, with earthworms and even sometimes some crabs or something like that. That could be also actually a testing ground for the cosmic zoo hypothesis in our solar system, if we were able to get there. Saturn's moon Titan, most amazing, because if there's life, it could be exotic life. Here's a radar image of the hydrocarbon lakes. And we work on one of the analog environment, Pitch Lake in Trinidad, where you have also a hydrocarbon lake, of course, a little bit different conditions, but we found their life being active as well. And Mars, of course, always very interesting. It used to be a water-rich planet, and now it's a desert. And if there's or was life, how far did evolution really go? Did it become extinct? Is it still there? And what can we learn from analog environments? So as conclusions, I like to put out that there's a predictable sequence of ecological transition in Earth's deserts with increasing dryness that also apply to Mars. If we want to find life on Mars, we have to follow the salt, not follow the water. We should investigate whether Earth is incredibly rare or whether it is they are more out like this. And this is especially interesting for a complex life, whether it's common in the universe. And future methodology like star shape will give us a first step in that direction. And we should also consider missions to astrobiological targets in our own solar system, such as Mars, Titan, and Europa, and the analog environments on Earth. I like to share this kind of picture over here, which I find very interesting. I got it sent by a student of Chris McKay. And this is a desert, and you see there's a plastic wrap from a sandwich bag that someone tossed in the desert. Of course, what you shouldn't do. But because of this, you actually have a microhabitat, and the lichen in it grows much better than in the surrounding area because it traps some moisture, and it's also protected from UV radiation. By concluding, I also like to say my thanks to the European Research Council that supported especially the research in the Atacama Desert and the one relevant to Mars. And if you like to know more, I have a couple of books out, like The Cosmic Zoo. I talked about it. I have also Life in the Universe. That's now in the 3rd edition, Expectation on Constraints. Cosmic Biology is this more popular science-oriented. Mega Catastrophes was a kind of a fun thing. What could all go wrong and how we get destroyed. And I couldn't help myself also to write a science fiction novel. So with that, I thank you and would be happy to answer any questions you have.
All right, thanks a lot, Dirk. It was great.
Thank you.
Yeah, thank you.
I should still point out, if you want to know more, also, I have a website, searchforlifeintheuniverse.com. Since a little time now.
Great, you found that URL.
Maybe I can ask a question.
Yeah, go ahead.
Yeah.
You made some very insightful observations just by studying one desert, Atacama Desert. Are there many other or several other deserts, analog environments for Mars on Earth that one should study? And how many are they? Is there any concerted effort to understand life on such a very taxing environments?
There are other analog locations studied in Morocco. There's a desert that is studied, and in Namibia, the desert is studied very well, I think, as well. My point would be that the best desert for as a Mars analog is really the Atacama, the hyper-arid core of the Atacama Desert, because it's the driest. I mean, when we look at it, we want to get closest. And also the dry valleys of Antarctica, they come very close to each of it has its advantages and disadvantages. But certainly we can learn a lot if we investigate other deserts as well, because as I showed, important is not only to be the closest to the Mars environment, but also some kind of succession. You can see how the ecology, how the organisms change when it's getting drier and drier.
Yeah. Yeah, okay, all right, thank you. I see a lot of creative books you have here.
Yeah, so I had a question. So when you say that follow the salt on Mars, some of these perchlorate salts, I mean, they are extremely harmful to organics. And so what do you think about that?
Yeah, we actually, one of my postdocs is investigating, you know, how much perchlorates different kind of organisms can take. And it's actually quite a bit. It depends still of whether it's sodium perchlorate or magnesium or calcium perchlorate. There are differences. But actually, Earth life, perchlorate are challenges, but can take actually quite a bit. We are still quite a bit away from that. What is, you know, what people calculate, how some of the under the surface, the brines also would be enriched in perchlorate. We're not getting there yet, or not yet. We're still quite a bit away from that. But we're looking for the salts. I mean, mostly the hygroscopic salts like sodium chloride. We know that even in a saturated sodium chloride solution, the halophilic organisms have no problems to survive. So, and I mean, with that, you get the water from the atmosphere. you have some protection from UV radiation and from ionic radiation then as well. And you know, I think we are sometimes a little bit too full of our kind of life on our planet. If there's life on Mars and it's still around and has endured in this dryness, arid desert climate for billions of years, they may have found some other adaptation mechanisms that are even more sophisticated than ours. So maybe they can, you know, I would expect can take higher salt concentrations and maybe can take higher amounts of radiation as well.
Right, right. There are perchlorate reducing organisms that you know about. So
yeah, right. Those exist too. Yeah, absolutely.
Any other questions? Raise your hand, type it out.
That was a wonderful talk anyway.
Yeah.
Yeah.
Yeah, if not, then let us thank the speaker. Thanks a lot, Dirk. And
Thank you.
yeah,
Thank
I
you.
look forward to reading your books and probably host you whenever all this craziness ends. So you can come here and explore this desert.
Okay, I would be happy to. Thank you for having me.
All right. Thank you. Thanks, everyone. See you all next week.
Bye.
Bye-bye.